Welcome everybody to this Drug Discovery World webinar entitled The Kinetics of Drug-Receptor Binding. Why is it important and how can we measure it? It is increasingly appreciated that the rate at which drugs associate and dissociate from receptors, the binding kinetics, directly impact drug efficacy and safety. However, the molecular determinants of drug-receptor binding kinetics remain poorly understood and are rarely measured early in the drug discovery process. Today we are delighted to have a panel of 3 experts from this field who will review current methods for measuring receptor kinetics and then describe some novel new approaches. So let's meet them. Firstly, we have Dr. Stephen Charlton, who is currently Professor of Molecular Pharmacology and Drug Discovery at the University of Nottingham here in the UK. He is followed by Dr. Louise Affleck, who is Business Development Manager for Life Sciences at Cease Bioassays. And then she is followed by Catherine Walk, who is a Senior Application Scientist with BMG Labtech. I'm Robert Jordan, Publisher and Editor-in-Chief of Drug Discovery World, DDW, and I'm going to serve as your moderator today. At any time during the webinar, you can send in your questions for our panelists by writing your question in the Ask a Question box and just pressing OK. The panel will try at the end to answer obviously as many of your questions as time permits, obviously we will try and get back to you if we cannot answer the questions within those times. So, okay, so I think we're now ready to go. So if you're sitting comfortably at your screens, let's get started. Our first speaker is, as I say, Dr. Stephen Charlton. So if you're ready, Stephen, the floor is yours.
Thank you, Robert, for that introduction. In this first talk, what I'd like to do is introduce the concept of the kinetics of drug binding and ask really the question of why it might be important, not only in terms of achieving better clinical efficacy, but also to better understand and interpret in vitro assay data. But before I launch into a discussion around kinetics, I want to introduce the team that's been working on this at Nottingham. The Molecular Pharmacology and Drug Discovery team moved to Nottingham about 18 months ago. We were previously working at Novartis Respiratory Group in Horsham. And when that closed down, we decided to move to Nottingham to join the already well-established cell signaling research group of Steve Hill. And because we've come from a drug discovery background, we have 2 main interests. Our key academic interest for a long time has been working in the area of kinetics, understanding not only the basis of kinetics of ligand binding, but also how that might affect signaling observed in cells. But we also have an interest in drug discovery, initiating our own projects, also working in collaboration to do collaborative projects or to support other people's internal projects in terms of assay development and detailed mechanism of action studies. So the first thing I'd like to do is really introduce binding kinetics. And really the top left-hand panel here shows us the simplest reaction scheme of a ligand with receptor. And we have binding according to the law of mass action. The rate of that ligand and receptor binding is driven by an association rate constant. And that has units both of time but also of concentration, because the association rate is concentration dependent. The dissociation rate of the ligand from receptor is defined by the dissociation rate, and that is only governed by time. Now, the ratio of the dissociation rate over the association rate defines the affinity at equilibrium, or the equilibrium dissociation constant. And we often use this measure as our measure of affinity, simply because the kinetics of binding are difficult to measure compared to saturation at equilibrium. The bottom left right-hand panel shows the association of a drug to a receptor. And you can see it proceeds over time and to a point where it reaches a plateau. Now, that plateau is the point it reaches equilibrium. That doesn't mean that the system is now static and that all ligands are bound to receptor. What that means is that the rate of association of ligand and receptor is equal to the rate of dissociation. So the system is still very dynamic. We have binding and unbinding events at that point. but we have no net gain in ligand-receptor complexes. Now I've highlighted two zones there. The first one in green is the equilibrium part that I've just described. There's also a whole section there in terms of time where the system is not at equilibrium, and this will differ depending on the kinetics of each compound. And I'll be coming back to this non-equilibrium situation in a few slides' time. In terms of measuring kinetics, I just wanted to introduce why we might be interested in it in terms of at least clinical benefit. Now, perhaps the best understood or the best well-known benefit is in terms of enhancing duration of action in the clinic. And certainly for receptors, I suppose the biggest example of this for some time has been in the inhaled muscarinic receptor antagonist field for the treatment of chronic obstructive pulmonary disease. There are two molecules here, both from Boehringer Ingelheim. The first generation ipratropium needs to be dosed 4 times a day, but the second generation tiotropium only requires once daily dosing. Now, although the pharmacokinetics are not identical, it was difficult for the team at Boehringer to really define why this was until they radiolabeled those materials and discovered that the dissociation rate from the M3 receptor, which is the target for these molecules, is much, much slower for tiotropium than ipratropium. And indeed, that 34.7 hours could contribute a large component to the once daily duration of action. And since then, I've been working with a colleague in Brussels, Georges Vauquelin, and we've done a series of simulations really looking at the impact of kinetics on duration of action. And certainly, you can extend the duration of action with a slow-off rate beyond the point where the pharmacokinetics means the ligand is essentially disappeared from the system. But the key point is here that this only really has an effect if the residency time outlasts the drug exposure in terms of pharmacokinetics. So if you have a very long pharmacokinetic profile, then off-rate plays no part really in the duration of action there, and that needs to be considered. So that's well established, but something that's probably less well known is the potential benefit for enhanced efficacy. Now, competitive drugs in an equilibrium situation will cause rightward shifts of concentration response curves to agonists with no reduction in the maximal effect in a shield type analysis. So this type of effect is surmountable, and it essentially means that if you put enough agonist into that system, you will always be able to outcompete the antagonist. But if we go back to that point where I described before, where we have non-equilibrium conditions, then this relationship falls down, and the compounds can appear to be irreversible or non-competitive, and this is termed insurmountable. An example of this is below for calcium signaling at the M3 receptor. Now here we have a concentration response curve to methacholine in the dotted black line, and we have that curve in the presence of 10 times Ki concentration of a variety of muscarinic receptor antagonists. Now you can see that all those antagonists shift the curve to the right, but they also cause a reduction in the maximal effect achievable by methacholine. And the degree of reduction there is directly related to the off-rate of those compounds. Now the reason this happens is, particularly in the calcium assay, is that it's a very rapid signaling system. And so between agonist addition and our measurement of our peak calcium response is often just between 2 to 10 seconds. Now in that time frame, if we have a slowly dissociating compound, it hasn't re-equilibrated enough to be fully outcompeted by the agonist. And so a number of those receptors remain blocked, even in the presence of a large concentration of agonist. And this is why we see this insurmountable behavior. Now this can have important consequences in vivo as well. So if we look across at the right-hand panel there, the graph, this is an in vivo experiment now looking at rat bronchoconstriction. We've induced the bronchoconstriction using an IV dose of methacholine, and on the y-axis we're measuring the lung resistance. And you can see as we increase methacholine, we increase lung resistance due to bronchoconstriction. But as we add our drug, this is given via directly to the lung, you can see not only do we shift that methacholine curve to the right, but we limit the maximal effect achievable. So you can see, even in a fully in vivo situation, we can see insurmountable antagonism. And this can have consequences not only for neuromuscular signaling, but also neurotransmission in the brain, anything that has a very rapid signaling profile. There's also good examples out there that in other systems, for example, the CCR5 receptor antagonists to treat HIV infection. And Pfizer had a very nice series of data showing that antiviral efficacy correlated very well with the dissociation rate of those compounds. So off-rate appears to potentially give us improved efficacy, but it can also have a downside. And a good example of this is in the dopamine D2 receptor antagonists used as antipsychotics for the treatment of schizophrenia. Now, the original typical antipsychotics have very slow dissociation. They cause quite significant extrapyramidal side effects. These are Parkinson-type side effects due to blockade of signaling in the nigrostriatal pathway. The newer atypical antipsychotics, like clozapine, don't have such an issue here. And it's hypothesized that these compounds have a more rapid dissociation. And because they're more rapidly dissociating, they display surmountable antagonism and therefore allow a small amount of signaling to occur, so don't completely block that system, therefore cause less side effects. So it could be that in some systems you might want to optimize rapid dissociation kinetics as well to avoid on-target side effects. But clinical benefits are not the only reason we need to understand kinetics. We need to understand the kinetics when we're actually characterizing compounds in terms of their pharmacological properties. Now, it's long been understood that if you don't leave an experiment, a binding assay, to equilibrate for long enough, you can either underestimate or overestimate the affinity of compounds depending on their rate of equilibration relative to the radioligand. And here we've got two examples of, again, muscarinic antagonists, tiotropium and atropine, which can either appear more higher affinity or lower affinity, depending on the time point. But something I really wanted to introduce, which is perhaps less well understood, is the impact on functional assays. Now, with functional assays for receptors, we have a number of different options to look at signaling. These range from very rapid signaling assays, for example, conformational change FRET biosensors that happen in the millisecond range, all the way up through calcium, cyclic AMP, to very long readouts, either reporter-gene assays or late-stage phenotypic readouts like proliferation of cells. Now, the key element here is that depending on where in that time frame we measure our response, can define where we are on our equilibration time point for our agonist. So a very rapid time point may mean that not all of our agonists has had time to bound to a receptor, so we may not have full occupancy. Whereas if we leave our system for much longer, we should achieve full equilibrium. And we've been exploring this functionally for a number of compounds agonists that have slow dissociation rates. The first example here is a compound called C26, developed at Novartis. This is a very slowly dissociating beta-2 adrenoceptor agonist, and you can see in the top left panel how it compares to a number of other clinically relevant compounds. And then we have three different signaling readouts: cyclic AMP, arrest and recruitment, internalization. And you can see that for each of those different signaling systems, that C26 has a slower signaling profile in the early time points than either isoprenaline or adrenaline, but at later stages, it actually appears to be higher efficacy. Now, depending on when we take our reading from, if we didn't, for example, do full kinetic reads, if we took the reading at an early time point, for example, where that green arrow is, you would call C26 a partial agonist compared to adrenaline. If you left the system longer and you looked at the red time point, you might call that agonist a superagonist. So the time point at which you measure these compounds can actually really define whether or not or how you define the efficacy of those compounds. And these data have just been published online now, if you'd like to read the paper in more detail. Another example was with a collaboration, again, that has just been published with Monash University, where we were looking at a series of D2 receptor agonists. And they had a series of lovely data looking at many different signaling pathways across different time points. And we identified that there's a compound, bifepranox, which has very slow association kinetics. And you can see in the functional readout on the top right there that while propranolol actually gets less active with time, presumably through desensitization of that response, bifeprinox actually increases its potency with time, so it behaves very differently. And in the bottom left-hand panel, you can see a measure of ligand bias. Now, I don't have time to fully explain how this is done. The paper has been published and it's highlighted below in Nature Communications. But the summary is that if you look at an early time point, this compound, bifeprinox, appears to be biased towards G proteins at the early time point, but then biased towards phospho-ERK and that pathway at later time points. So what this tells us is that if we're trying to characterize compounds, we need to understand their ligand kinetics before we can really start calling compounds bias or really denoting their efficacy. So if kinetics are very important, how do we measure them? Now, in terms of drug discovery and thinking about this, what I've done is put a standard drug discovery scheme here, starting from screening through to hit validation and lead optimization, and then finally on to development. And at the top of that, I've just taken a very broad view of how many compounds might be tested at each of those stages. Now, kinetics traditionally has really only been measured using radiolabeled material, which tends to only be available during late-stage drug discovery, where you have one or two selected compounds. And there the kinetics can be investigated using standard filtration type assays or SPA assays. But that, of course, although you can characterize your clinical compound, it doesn't give you any information on allowing you to develop the kinetics per se as part of the optimization process. The advent and the introduction of surface plasma resonance techniques has really revolutionized our ability to look at more compounds, particularly for soluble proteins such as enzymes. There's still some issues in utilizing this with membrane proteins, and some compound classes also have issues in terms of interacting with the chip. But certainly we can look at hundreds of compounds using this, and I do know some groups who actually run thousands of compounds through these systems, although it takes a huge amount of effort and multiple machines to do that. But what we were really looking for is a method that would fit into this hit validation phase, where we could look at thousands of compounds in kinetic mode. What we wanted to achieve during this assay development was achieve a 384 well-compatible system. We wanted our receptors to be in our native lipid environment, ideally in a membrane, and certainly if we were able to look at in whole cells would be perfect. We wanted a system that was able to have a continuous read detection, so we were able to look at kinetics in a single well. And it was important to us that we had an injector system, because we needed to look at time points very early on after compound addition. And finally, we've done a lot of work previously looking at the influence of temperature on kinetics, so having full temperature control was also important. So really, with all those in mind, what we did is we looked at a variety of different systems out there, and we settled on testing the Sysbio Taglite assay system in conjunction with the BMG PHERAstar, which in our minds fit these requirements. So what I'm going to do now is stop there, and I'll hand over to Louise and then Catherine to talk through these systems, and then afterwards I'll come back and talk about our assay optimization phases. So I'll leave it there and hand over to Louise now.
Thank you, Stephen, for that fantastic introduction into the Taglite receptor ligand binding system. Taglite is a fluorescent cell-based assay format enabling ligand binding kinetics and ligand binding affinity determinations, whereby a G protein receptor is expressed at the cell surface and a SNAP tag is expressed at the N-terminal of the receptor. The terbium is then covalently labelled to the SNAP tag. The ligand to the receptor is then labelled with the acceptor. When the labelled ligand comes in close proximity to the donor molecule, the terbium, a FRET event is generated. Unlabeled ligands, unlabeled test compounds can then compete off any labelled acceptor present, thereby preventing a FRET. Taglite is easy to use, where the labelled cells are plated out and then the unlabeled compound of interest is added, followed by the labeled fluorescent ligand. As mentioned earlier, it's a competition event, so you'll measure a decrease in the FRET signal in the presence of unlabeled compound. The BMG FX reader is an ideal reader to detect the HTRF FRET signal, and Catherine Walk will later describe this in much more detail. So to wrap up, the TagLight system enables ligand binding kinetics due to the no-wash homogeneous assay format. It is a non-radioactive, fluorescent and robust signal is generated. I will now pass on to Catherine Walk at BMG LabTech to describe the PHERAstar FSX in greater detail.
So thank you very much, Louise. My name's Catherine Walk and I'm an Applications Manager with BMG LabTech UK. I will be talking today about the PHERAstar FS instrument used by Stephen in his research to date. I'll be covering the standard features of the instrument, but paying particular focus to those that benefit the kinetic TagLight approach. I will also take an opportunity today, though, to also introduce our latest product innovation, the PHERAstar FSX instrument, the new gold standard for HTS. The PHERAstar FSX, for those interested in the kinetic TagLight approach, will offer also advantages over the PHERAstar FS system, so it's worth noting in this presentation. The PHERAstar FS was introduced in 2009 and is now a very well established solution for medium to high throughput screening, with its design features made to accomplish both high sensitivity and speed for all plate formats up to 3, 4, 5, 6. Key to the success of this instrument has been the use of dedicated read technologies for all read modes, with high energy xenon lamp used for optimal fluorescence excitation, a dedicated channel for luminescence, an ultra-fast spectrometer for absorbance, and of course laser options for both alpha technology and, most importantly for this talk, a TRF laser for HTRF. The system also includes a standard simultaneous dual emission for all read modes mentioned on the slide, fluorescence and in particular TR-FRET, as is relevant to our talk today. So, the PHERAstar FS is an easy-to-use system and utilizes an optic module concept. These barcoded modules contain all components needed for an assay, as with the HTRF module that would be used by Stephen's team. All filters, dichroic mirrors are fitted inside to avoid error in installation and to protect these components. Important for HTRF is our use of dedicated redshifted photomultiplier tubes, as these are photon counting detectors and they offer the best sensitivity in this application. And important, finally, to the TagLight approach, as Stephen's previously mentioned, would be the injection and incubation system, also available for the PHERAstar FS. Other standard features that benefit FS users in and around this GPCR area are obviously top and bottom reading capability. This is controlled within the software, making it possible to read even the cell-based TagLight assay either from top or bottom with no user changes. Bottom read may be employed where using monolayers of cells. The advanced Z-height detection, possible from both top and bottom, is not a general feature offered by most HTS units, but it does ensure the best performance in reading in either orientation. Finally, to make this system more ultimately suitable as a screening tool, the instrument includes plate barcode readers capable of reading west, south and east, and also has a stacker option with a continuous loading feature. So now I come to technologies that have maybe additional impact or can help with setting up the kinetic TagLight assay. I've already mentioned the injectors and incubation setup. I can focus a little bit more on the injectors shortly, but we also have high resolution wire scanning as shown in the image at the bottom of the page. And also that's useful for looking at receptor expression levels. And also on the right-hand side, you'll see a decay curve monitoring curve. This is useful if perhaps optimizing timings or looking to improve assay sensitivity. So now I come on to the specifics for HTRF. We offer two light source options for exciting HTRF, a xenon flash lamp and a TRF laser. As you can see from the data shown on the right-hand side produced by Cisbio, the flash lamp and laser both perform well with great Z times for both europium and terbium donors, but the laser that is optimized at 337 nanometers has a little bit of an advantage with terbium-based donors, creating up to 30-35% increase in signal window as seen by the data. More importantly, for the tag light application, the more energy supplied with each flash of the laser as compared to the flash lamp makes it suitable for reducing flash numbers, increasing read or decreasing read times for both 384 and 1536 to achieve the fastest read times. We have real flexibility with our injectors. Users can add changeable volumes from well to well and add volumes as little as 1 microliter, changeable in 0.5 microliter increments. Speed of addition can be regulated by the user to prevent cellular damage, damage to the cell layer. And the most important point is that with the injector system sitting up directly above the well, we can inject and read simultaneously to ensure that we get the fastest dispense and read times over the plate, which is important when trying to sample at a high kinetic rate. Okay, so I mentioned I would come on to talk about the PHERAstar FS system. We have built on the success of the PHERAstar FS with this platform, and I've looked to re-engineer all of the optical components to gain sensitivity advantages in all of the major read modes. We also have faster read times for TR-FRET with a new faster laser, broader dynamic range and luminescence, and have introduced for alpha technology enhanced performance and also simultaneous dual emission for alphaplex. For time-resolved fluorescence performance, I've previously mentioned that we've introduced a new, faster laser system. The increase in laser speed would make it possible for Stephen to accomplish two possible outcomes. His team could continue to use the existing fast capture speeds, but increase the flash number by 50%, which would result in higher signal counts, or they could continue to read with the same number of laser flashes and expect to see an improved plate read time by up to 33%, as shown by the data on this slide. I have two slides that exemplify this with respect to 384 and 1536 well plates to show you. The red line on the graph shows the drop in read time by using the same number of flashes, and the green line shows the gain in flashes at the same read time for a 384 well plate. We have read time on the y-axis and laser flashes on the x-axis, and also for 1536 as shown here, to give you an idea of the plate read times. Now obviously for Stephen's application, speed is very important. So I would like to finish off with a brief summary. Both the FS and FSX instruments are perfect to handle this rapid read kinetic HTRF application. The laser and injectors are both important factors for the accurate determination of on and off rates. And the new PHERAstar FSX, whilst offering general HTRF assay performance improvements with its flash lamp, also with the addition of the TRF laser, will give further benefits to the tag light application with respect to speed of reading or higher counts where as needed. Finally then, just to acknowledge our team in Germany and also the Cisbio team that have been helpful in preparing this data. We hope that you will put us to the test as Stephen has, and at this stage I pass over to Stephen for him to show some of the data that has been created using our the tag light and the PHERAstar system. Thank you.
Well, thank you very much, Catherine. I'm going to finish now by talking about an assay development utilizing both Cisbio and PHERAstar to get a kinetic association assay using HTRF. So really to refresh what our requirements were, again, I've talked this through already, but we really wanted something that had a continuous read in a 384 well format. The systems, as I said before, we're utilizing tag light and PHERAstar. What I want to highlight on this slide is that we utilize the beta-2 adrenoceptor as our model system, and the tracer ligand that we have is a fluorescently labeled propranolol. Now, the first thing that we needed to achieve is the fastest read time possible. Now, the reason we need this speed is because the way we're formatting this assay is to add our reagents into a well and start and take a read immediately then. Then what we do is move to the second well, add our reagent and read. And we need to do that to as many wells as possible before we then go back to the first well again and take the second read on that first well and then move back over that line. So we're rereading a number of wells at any one time. Now, the faster we can read each one of those wells defines either the number of wells we can read in one plate, or the time difference between each reading for each well. So for very rapidly equilibrating compounds, we might need very short time reads to enable to fully fit our kinetics accurately. So the first thing we wanted to do is understand how we were going to excite our donor. And we had two options with the PHERAstar, either laser or lamp excitation. What I've got here on the two top panels are a saturation curve to our fluorescently labeled propranolol, either using the laser or the lamp. And this is the emission at 665 nanometers. And what we have here in the different curves is different numbers of flashes of either the laser or the lamp. Now, you can see immediately that the laser is higher power, higher emission, and we get a much higher signal with far fewer flashes. If you ratio those numbers, they come out very similar, as you would expect. But what we wanted to do is really compare whether this higher overall window would give us better reproducibility and less error around repeated measurements. So what we did is we took 52 wells, all identical wells, in the center of a 384 well plate, and we read them in succession using a different number of excitation flashes. And you can see the number of flashes along the x-axis there. And then we have our error, our coefficient variation, on the y-axis. And we were aiming below 10%. Now you can see, if we look at the blue, that with our initial amount of flashes, up to say 10 or 30 flashes, we started to get close to our target of 10%. But with that number of flashes, it was taking us really 30 seconds to read those 52 wells. When we moved to the laser, already, even just a single flash, we were getting less than 10% variation across those 52 wells. And if we used 3 laser flashes, we were improving that still. Now, the benefit of using the laser is that we can get across those 52 wells in just 4 seconds using a single flash in scanning mode. And so we felt that the laser gave us an opportunity to get much more rapid reads. And so we utilized the laser from this point onwards, either in a single flash or with 3 flashes. So the next thing we really needed to do is to characterize our tracer ligand, our fluorescent propranolol. And in the first panel here, you can see the dissociation kinetics, the association and dissociation kinetics of that ligand. We've added the ligand and watched it dissociate over time. And then at about 25 minutes, we've added a large concentration of competing compound, and we can see the dissociation phase. All of those data were generated from a single well. The second panel looks at the association of different concentrations of propranolol. And this shows that the association rate is concentration dependent. And we can globally fit those data to a model of association to generate K on and K off. And you can see that the numbers are fairly similar across both. But again, from that right-hand panel, we only have 4 wells from a 384 well plate generating all those data. But what we really wanted to do is to really measure the kinetics of unlabeled compounds. And to do that, we employ a competition association assay format. Now, this was first really described, at least in theoretical terms, by Matulsky and Mahan in 1984. And it involves addition of both the tracer and unlabeled test compound to the receptor preparation at the same time. And then the association of the tracer is monitored. And the association rate of the tracer will change in the presence of an unlabeled competitor. What we're able to do then is use this mathematical model that they described to fix a variety of parameters that we've already fixed in our experiment. For example, concentration of ligands. We know our association and dissociation rate of our tracer. And if we fix all of that, we can then globally fit our association rate and dissociation rate for our unlabeled compound. And you can see on the bottom of this slide, we've utilized this methodology using radio-labeled methods over many different years, over a variety of different systems. And those are just some of the papers that we've published in this area. And you can go and learn a little bit more detail around this methodology. But for the HTRF, the first experiment we did in this competition mode is highlighted here. And this is just one of the examples. So we've got salbutamol, which is a beta-adrenoceptor agonist. In the left-hand panel, in the blue, you can see the association of fluorescently labeled propranolol alone, and then how that is altered in the presence of increasing concentrations of unlabeled salbutamol. And the data you can see in the table below that we achieve by fitting that is fairly similar to that we achieve using our standard radio-label assay. Now, something I want to draw your attention to here is the difference in throughput. So our radio-label experiment, we had 3 competitor concentrations, and that took a whole 96-well plate to generate those data. On the left-hand panel with the HTRF tag light system, we used twice as many competitor concentrations, and it took us just 7 wells of a 3 or 4 well plate. So you can see a large improvement in throughput there, and also in reagent saving. Now we ran in our test case a variety of different beta-adrenoceptor ligands, and here we're comparing the on rate and the off rate measured from the HTRF versus our standard radioligand assay, and you can see that there's a fairly good agreement there across those 2 systems. Again, though, I'd like to draw your attention to the fact that each end for the above experiments required 25 wells using the tag light system, but 768 wells using our standard radioligand binding assay. So we're able to generate those data in a much more rapid manner. A question we asked ourselves really was how many concentrations of competing ligand did we need to get accurate estimates of our on and off rate. And I'm showing the same data here for salbutamol. But what I've done is at each point, each one of those curves reaches equilibrium, which is the plateau point, we can generate from those data an IC50 curve that I've shown on the right-hand side. Now what we did is we globally fitted all those data, as I've shown you before, and then we just globally fitted the control curve, but just the single concentration of salbutamol, which is the lowest concentration we tested. Now that arrow denotes that there was less than 20% inhibition on our IC50 curve at this point. But when we used that single concentration alone, the fit that we got was not dissimilar, and certainly in terms of the equilibrium constant, the PKD that we estimate there, was similar to that achieved by using the whole concentration range. And this is perhaps our first signal that we might not need to run a large number of concentrations, and we might be able to generate on rate, off rate, and equilibrium dissociation constant values from single competitor concentrations. So that was really our first foray into this system. That was our test case. We then went on to really apply it to something that we were really interested in. And this goes back to our question around the D2 dopamine receptor field. Now, I've explained to you our interest in this, in that the typical antipsychotics appear to have slower dissociation. The atypicals with lower side effects have a faster dissociation. But the studies from which these conclusions were drawn range across several decades, several different labs using different technologies, different assay conditions. So it's very difficult to compare all those data in one go. So what we wanted to do is utilize this high throughput system to look at a large number of clinically used antipsychotics to measure their dissociation rate and association rate, and to see if this hypothesis holds true. Now, when we were generating this D2 receptor assay, we utilized snap-tagged D2L receptors expressed in CHO cells, and those were generated by Nick Holliday at the University of Nottingham. The tracer ligand we used was a fluorescently labeled PPHT, which is a partial agonist of the D2 receptor, and that was provided by Thomas Rue at Cisbio. And throughout these experiments, we used 3 laser flashes on the PHERAstar. Now, the first thing we did is characterize our tracer, PPHT, and here you can see at the top we have a standard saturation binding analysis at equilibrium, and I really wanted to show you here the non-specific binding levels are very low using this system, even at relatively high concentrations of ligand. And then the panel below, we show an association kinetic assay of the tracer alone at different tracer concentrations, and you can see that very nicely obeys law of mass action binding to a single site. And from that we can derive our on and off rate to take through to our competition association assay calculations. So if we move on now to the experiments we did in the presence of unlabeled antipsychotic compounds, here's an example of some of those data. In panel A, we have the IC50 curves generated from the equilibrium point of each of these experiments. And then we have 3 examples of compounds that show different types of kinetic parameters and different profiles in this system. And from this, we were able to generate the on and off rates for these compounds and really correlate them with the affinity constant, but also to look at whether or not they were related in terms of their side effect profiles. So in this top panel here shows the on rate on the left-hand side and the off rate correlated to the equilibrium affinity constant. And an interesting observation is that for the atypical antipsychotics, the on rate appears to be quite well correlated to their affinity. But for the typicals, that's not the case. For the typicals, it's the off rate that appears to be driving their affinity. So that's an interesting potential distinguishing feature. But the real question as to whether or not the typical compounds have a slower off rate and therefore generate a more profound blockade of that system and therefore generate extrapyramidal side effects, when we looked at that question, we found that although some of those typical compounds had very slow dissociation rates in the green, there was a significant overlap with the atypical. So there are a large number of typical compounds in there that show side effects, but which have off rates that are very similar to the typical compounds, which don't show so many side effects. So although we have some support for this kinetic hypothesis, we've also identified that it can't be the whole story and that there's probably other contributing factors in this. We've since done a much more full study and correlated these with clinical readouts as well, and we have a paper that's submitted which hopefully will be through the review process soon and available for download. So I just wanted to, before finishing, just go back to this question of enhanced sensitivity compared to IC50 assays. So we use the same type of analysis that I showed you before for the beta-2 adrenoceptor with this dopamine D2 receptor system. And here is an example for propranolol. So we have our different concentrations of propranolol, and again, each one of those curves is generated from a single well. We fitted all of those together in all 6 concentrations globally fitting. And then we've just compared that to the single concentration that gave us the lowest inhibition, so less than 25% inhibition of our tracer binding. And you can see on the right-hand panel, when we compare the equilibrium dissociation constant estimates from both of those, whether we use all of the concentrations in that fit or just the one that causes less than 25% inhibition, there's a very good correlation of our affinity estimates. Now what this means, firstly, is that we are able to predict affinity constants and on and off rates from fewer numbers of competitor concentrations than you need to generate a whole IC50 curve. But importantly, we can by fitting this whole kinetic curve at once, we can generate data for compounds that cause almost no inhibition at equilibrium. So this allows us to look at much lower activity compounds and potentially opens up this system for looking at low affinity fragment screening for GPCRs. And this is certainly something that we're following up on now with a couple of test cases. So to revisit my initial question, what we were aiming to do, I believe that we have established now higher throughput assays for receptors that allow us to look at the kinetics of unlabeled compounds, successfully using the tag light combined with the PHERAstar to generate this system where we can get kinetics from a single well of a 384 well plate. I put at the bottom there that I think that really this now supersedes our need to do full IC50 curves, and indeed we get more information, we get the kinetic information out of this as well as our affinity constants. So I think this could essentially replace generating IC50 curves during the HIT validation phase. So to conclude, I guess across both presentations, I hope during the first presentation that I gave examples of why I think kinetics can be an important element to look at in drug efficacy and safety, and really that slow off rate is not always better, and that the type of kinetics that you try to optimize will be very dependent on the physiological system that you'd like to modulate. And then the second presentation shown that we've managed to establish higher throughput kinetic FRET assays, which allows the assessment of kinetics at a much earlier phase in a high throughput mode. They're more sensitive and efficient than IC50 assays, and again, just to say that I really believe that this will open up our ability to do fragment screening in kinetic mode on GPCRs. I wanted to say with the paper there, this is the first paper that's been published by us using this method in Nature Communications, and this is the study I talked to you about previously in collaboration with the MIPS Monash team with Rob Lane and Arthur Christopoulos. And we have several more on the way. So finally, I just wanted to end by acknowledging people who did this. The person I need to acknowledge most is David Sykes. He's done a large amount of the work on the CysBio tag light system, and indeed has been working with me for many years on these competition association kinetics assays. There are other people at Nottingham who have assisted here. Obviously, the collaboration with Monash University has been excellent with that Nature Communications paper. George Rouquelin has helped do a lot of simulations and modeling over the years. And of course, the teams at both BMG LabTech and CysBio for their assistance and support while we generate these assays. So that's the end of the assay development phase. I'd now like to hand over to Robert, who will open this up for questions.
Well, thank you, Stephen, and thank you to all of our contributors. I thought they were all very valuable in their own right. Okay, we now have actually plenty of questions, but before we start our Q&A session, I'd just like to let everybody know that this webinar will be stored on the Drug Discovery World website, and it'll be free, available on demand. So the site will be www.ddw-online.com. Give us a few days to get it up there, but it will be up hopefully sometime next week. Let's go to our questions, and I think the first one we are going to ask is from Benoit Fouchac. I hope I pronounced that properly, from Eurofins Pharma Discovery. And he's asking, and probably this is to you, Stephen, what is the influence of the tracer's affinity on the kinetics, and what is maximum ratio of affinities, i.e. tracer versus competitor, that is acceptable?
Right, okay, thank you very much. So I think that this was one of several questions. I'm asking a similar comparison between the kinetics of tracer and competitor. The kinetics of the tracer certainly affects the accuracy of your kinetic estimates that you get from this type of analysis. We find that if you have a very slowly dissociating tracer, then accurately ascribing kinetics for very rapid competitors is very difficult, and there is a point by which you cannot distinguish the kinetics anymore. So we've actually spent some time trialing different tracers to look at their kinetics versus compounds with different affinity. We're actually at the moment in the process of finishing a paper describing this using both simulation data and also experimental data that we've used around the dopamine D2 system. So that will be out there. I don't think it's as simple as to give a simple ratio of affinities right now, but I think that certainly if you look at some of the simulations that will be in that paper soon, then that will give an indication of the type of ranges that we'll be looking at.
Good. Thank you, Stephen. This one is from Ying-Kai Wang from Bristol Myers Squibb, BMS, probably directed towards you, Kath. How does the PHERAstar reader compare with PerkinElmer's Envision or ViewLux? Kath, are you still with us?
I'm here.
Okay,
sorry,
did you hear that
sorry
one?
there, Robert. I did pick that up. A little bit of a malfunction there with the mute button. Apologies, everybody. I won't take too much time away from the technical questions. The PHERAstar FS and FSX have probably some of the best specifications for drug discovery with respect to all read modes. For the time resolved fluorescence TR-FRET application that Stephen's talking about here, the fact that we use photon counting devices means that we can get down to very low levels of signal. That in combination with the TRF laser means we can get a fantastically high signal in a short space of time. So in comparison to the Envision that uses a standard integrated photomultiplier tube or the ViewLux system, we should be able to see hits that perhaps those systems wouldn't be able to see, and we certainly for this application should be able to achieve a very high signal in a very rapid read time. So have a look at the specifications. Please contact us if you want more information on the PHERAstar or would even like to try one in your lab. We'd be happy to help.
All right, excellent. So this one, Michael Bradley, Cyrus Pharmaceuticals. Probably, this might even be you, Louise, I'm not sure. What acceptor fluorophore was appropriate for use with the TB-labeled SNAP-tagged protein?
It's really dependent on the GPCR in question. For some GPCRs, we found that the red acceptor has been better, and for other GPCRs, we found that the green acceptor has been better. So if you look on our website, you'll find the list of ligands that we've already labelled and we've already optimised as well, so whether they're red or whether they're green.
Excellent. All right. We've had a few, some questions, some statements. This one's from Professor David Calhoun, I think I pronounced that correctly, from UCL, who's saying that the association rate constants you get seem surprisingly low. Usually we get 10.7 or 10.8 per second, not far from diffusion limit. Obviously one for you there, Stephen.
Yes, and thanks to David for the series of questions. Very technical, and I should point out actually that David is one of the really early pioneers in looking at kinetics and certainly competition kinetics. So I think the examples I gave did have slow association rate constants. We do see for agonists that they tend to be slower than antagonists. When we look at antagonists, we do see rates in the range that David stated there. But I think this really could speak to another concern that David raised in another one of his questions about the use of this simple single site analysis to look at agonists. Now, the thing that agonists do is they can change the conformation of the receptor, so you potentially have multiple sites that you're looking at. We try to limit that as much as possible by including high concentrations of guanine nucleotide to drive at least away the G protein dependent stabilization of a high affinity state. But there's no doubt that even in the absence of an effector, there is probably some isomerization step happening here. So I think we need to be very aware that the kinetics we're measuring here are a global rate constant. There's probably a number of more microscopic rate constants embedded in that we can't detect at the moment for GPCRs. Although certainly for ion channels and a lot of David's work, you can start to drill down into some of those more specific rate constants for each isomerization step. But certainly for receptors right now, we have trouble teasing those apart. So I think we need to be fairly pragmatic when we look at these numbers. But I think that I think there are two components controlling the association rate here. One for agonists is potentially this isomerization step that might be occurring. The second, I think, is something that I'm quite interested in, and that's that the association rate is largely driven, well, is driven also, at least the estimation of the association rate, by the concentration of drug that you have in the system. Now, we assume that the concentration of drug that the receptor sees is identical to that that's added into the well, into bulk aqueous phase. But some work we've been doing recently using fluorescently labeled compounds and fluorescence correlation spectroscopy methods has shown that the concentration actually close to the receptor itself, within the membrane region or just above the membrane, can be very different to that in bulk aqueous. So I believe that when we're measuring these rate constants, there is a component of non-specific local concentration of compound that's largely driven by the physicochemical properties of that compound. So I think we need to be careful when we're interpreting association rate constants. So that was a very good question, and I think that what it shows is that certainly for GPCRs, we're not there yet. And we've got a little way to go before we can fully tease out all these microscopic constants.
Excellent. Thank you, Stephen. And Stephen, can you help Kirsty Bennett out from Heptares Therapeutics? She's finding at the higher red agonist concentrations binding is higher at very early time points compared to later time points. Can you explain why this might be?
I'm afraid I can't. We have done red agonist binding, and we don't see that. So I think, Kirsty, the best bet there is to maybe just send me over some data, or just maybe give me a call, and maybe we can talk about your specific question there. It would be easier for me to see the data to answer that.
Good. All right. I'll leave you to sort that out between you. Question from David Green at Vertex. Are there any issues around the overexpression of tag-like receptors relative to endogenous receptor levels? In other words, are you going to see inverse agonist type effects due to overexpression of receptors affecting the stoichiometry with coupling proteins and GPCRs?
Are you directing that to me again?
I've no idea. I think probably yes. I think it probably is that for you, that one, Stephen.
Okay, that's there are a fair few points in there. I think that in terms of just the simple overexpression question, the good thing about these time resolved FRET systems is that it's a very specific effect. So we're only looking at binding activity to a tagged receptor. Now, if you have a lot of other receptor in there, it could complicate issues in terms of potentially depleting high affinity traces. So if you get a lot of binding to endogenous ligands, you may reduce the overall concentration of ligand available to bind your tag-like receptors. But also, I suppose, if we're thinking about what we spoke about before with agonist binding and maybe a dependency on effector proteins, then I'm sure, of course, that if you have two receptors competing for the same effector, you may have a competition issue there. I think we simply don't know enough. I think the only real way we've looked at this at the moment is looking at G protein dependent high affinity binding with agonists that I described earlier. Although there are now examples of arrestin dependent stabilization of particular receptor states as well. These are all very good questions that I just don't think are well understood in the field yet. And I think that there are other systems. There are people using Brett systems where they're using, rather than these two fluorescent ligands, they're using luciferase to measure out and looking at receptor and effector coupling as well. So those systems in combination perhaps with these FRET binding assays, we might get closer to that, but I don't think there's one straightforward answer for that question at the moment.
Okay. All right. Look, Stephen, Louise, Kath, thank you for those answers. Ladies and gentlemen, unfortunately, we are about to run out of time. There are an awful lot of unanswered questions that you've put up there. What I'm going to do is send these questions to our speakers, and then hopefully they will get back to you via email, and you can talk it out between you. Let me just remind you that this webinar will be available online, ddw-online.com. I'm free on demand. Give us a few days to get it up there, and we will be away. I just thank you, the attendees, for obviously attending, and hope you'll join us again for another DDW webinar. And thanks once again to our speakers and our sponsors, Cease Bio and BMG LabTech. Thank you very much, and goodbye.